Hi guys, welcome back. Go back to another week. Hope you're all doing great. Getting pretty deep into the semester now. How are you all doing? Hello, hi. Good to see you all again.
Hi, Luke.
Pure vibing. Oh, great. Let's keep the vibes going. All right, so looking at EEG this week. Very cool topic. Really common in our unit at UTS that I'm in. We look at all kinds of cool stuff with EEG, other areas of electrophysiology in general. I apologize to any of you who are doing currently medical and applied physiology because this may overlap a little bit. It will definitely overlap a little bit. So David Van Rijk's subject is looking a lot at different types of electroencephalography. So you'll do like ECG, EMG, and EEG as well. So looking at kind of a pretty good range of electro sort of physiology in general. In this subject, we're just going to be looking at EEG. So just looking at electrical activity of the brain. Obviously, it's neuroscience. It's a really important aspect of it. It's fine means I have to learn less things. Yeah, that's true. I'm not sure how far you guys might be, those of you who are doing MAP, how far you are through this. Be interested to know those of you who have actually already done the EEG component yet in the subject. I'm not sure yet. But I have helped David prepare some of the content for MAP. So yeah, you'll definitely see a little bit of overlap, but we'll try to keep it minimal. We go pretty heavily into like how individual neurons function and how they work as populations, and really like get into like what an EEG is and how it can be used for like diagnostic tools for like sleep disorders and epilepsy in particular is what we're going to go through today. Anything we should prepare for the prac exam? I assume you mean your anatomy exam? Not sure, but for EEG, not really relevant for the prac exam. So we're just going to jump straight in, and we're just going to start with something like sort of generic like summary or background introduction into this. So an EEG waveform, I've got a couple of examples on the right hand side of the screen. So what an EEG is basically looking at large populations of neurons beneath the surface of the skin that we don't want to have to like probe someone in their brain to be able to read those neurons. So what we do is instead is that we just stick a electrode to the surface of someone's skin, and we are just kind of hoping that things like bone, muscle, skin, all that stuff that's in the way, just hoping that that's not getting in the way of this signal that we're trying to read. And that's common across all areas of electrophysiology. Whether you're trying to measure muscle activity or brain activity or like cardiac activity, we, as like an easy way, as like a non-invasive way of measuring these activities, we just use contact with the skin. And there are some problems with it, but it's really, really good for the most part because it means you don't have to like cut someone open and probe them with sort of invasive tools to get a reading of these cells. So a single neuron is capable of generating many, many action potentials per second. And an EEG waveform is the result of lots and lots of neurons firing at different points rapidly. And we might expect an EEG to therefore resemble one really large action potential, but that would only be the case if it was all like 1 million neurons all firing at the exact same time. The result of that would be just sort of one big, like it would look like one enormous action potential on an EEG. That's not the case. That is a marker of an extremely unhealthy state. What actually happens is that neurons do not fire in sync, so they're asynchronous and they activate at different times. And this causes their electrical potentials to kind of overlap. And if you guys remember from sort of physics a little bit, when you have two waves that overlap and they don't, sort of one of them is going high at the same time as that one's going low, they actually cancel each other out. And so you end up with a smaller amplitude than what you would normally get if they were both like contributing to each other, if that makes sense. So this is what causes your like a waking EEG, like a healthy EEG, to have a really low amplitude or height. Remember, amplitude means like the height of a wave, but it means that they have higher frequency. So frequency is like the narrowness of the reading. And if you just look at some of these brain waves on the right-hand side, you've got in your relaxed sort of wakeful state, you have, oh, so that's eyes closed, so that's slightly different. Let's look at your awake or excited. So this looks quite chaotic, this second one here, but this is what a waking EEG looks like. When you're doing a little bit of mental work, a little bit of maybe physical work as well, this is what your beta waves, for example, look like. So really small, but really high frequency, and they look quite chaotic. But it's very healthy. It's a very healthy looking EEG. And then when you're looking at like slow wave sleep or deep sleep, for example, this bottom one, that's where you have less work going on in your neurons. And so the resulting EEG waveform has these really big amplitudes, but really smaller frequency. And we'll talk a little bit about frequency in the slide after this one. So this slide is just to further explain how this works. If you look at B, you've got 1, 2, 3, 4, 5, 6 individual neurons firing, each of them sort of having different levels of electrical activity, which makes sense. And then at the bottom, you have the sum of their EEG. So what would happen if you had an electrode that was picking up on all 6 of the signals from these 6 different neurons? So as you can see in the top one, if you have an irregular or asynchronous EEG, it looks something like this. And you can see that if you look at any random point, the sum EEG is kind of being cancelled out by the various different electrical activities at each different point. It's easy to see in this bottom one. You can see if you synchronize the firing of all 6 neurons in this sort of first instance, you can see that the sum EEG will also represent that. And that's basically what we mean by synchronous versus asynchronous activity and how that might look on an EEG. All right, so the first question that we're going to look at comes straight from your lab manual, as do most of these things. So you need to be able to recognize the 4 main EEG activities that we look at in this subject. There are a couple of other ones, but these are the 4 important ones that you need to know. So you need to be, this is a really good table, one, because it gives you the range for each of them, and it gives you your behavioral or psychological state where you would expect to see these. So if you're kind of compiling like a bunch of notes, like a couple of pages of notes that you want to have in front of you when you're going through your exams, highly recommend something like this, because this is a whole bunch of different questions that we can ask you. For example, so starting at the bottom with beta is your awake state or your alert cortex, and it's common during mental and physical activity. Going back up to, and it has very high frequency. So frequency is, when we talk about EEG, we talk about in terms of hertz as a measure of electrical activity of your neurons. And then if you move up into alpha, so now we're getting into slower frequencies, so between 8 and 13 hertz. But generally we're talking about larger amplitude or larger height of the wave. And it's just coming back to what we talked about in that previous slide again. So your alpha waves are more prominent during like a relaxed state, when you just kind of got your eyes closed and you're just sort of in a chill state. And your alpha waves will be disrupted by any kind of mental or physical work. So you really have to be kind of not asleep, definitely not asleep, but when you're just sort of in a Zen state, just like maybe just chilling basically. Yeah, it doesn't represent any kind of mental work. So moving into your slow waves now, you've got your theta and your delta waves. So theta are between 4 and 7 hertz. They're associated with some sleep states and drowsiness and fatigue, as well as rapid eye movement sleep, which we go through a whole lot when we do sleep and dreams, problem solving, hypnosis, and meditation. And then you have your delta waves. So these are the slowest waves. They are less than 4 hertz. And as we saw on that previous slide, in your first slide, they look like this one at the bottom, really, really sort of like almost mountains. And that is representative of, if you saw someone with an EEG like that, you would assume that they were in a sort of slow wave sleep or a deep sleep. Cool. And so we can study these different brain EEGs. They're really useful for studying if we're looking at specific pathologies. Epilepsy, it's really important in a lot of sleep and behavioral studies. It's really important in. And the great thing about EEG is that you can just attach these electrodes to someone's head. They're not protruding into the skull or anything like that. It's literally just like a metal cap attached to someone with a little bit of gel on there, and that's wired up to a computer. And so you can have someone in a sleep lab where they stay overnight, maybe even up for 24 hours, but usually just like for up to like 10 hours while they sleep. You can study their EEG patterns, and you can make various different types of diagnoses based on that. So EEG is very cool. This slide is mentioning a couple of important things that, we go into things like spindle bursts when we do sleep. So I wouldn't worry too much about this. This is just giving you more like examples of like behavioral and psychological states where you see different characteristics of an EEG waveform. Okay, the way that we measure EEG, we do it based on what's called the International 10-20 system. So this is a way of sort of breaking down the brain into different percentages based on 10 and 20% of the circumference of your head. So the reason we do this is because ratios, 10 and 20%, even 5% sometimes, ratios are better to use, or I guess they're more standardized than just saying, all right, well, if you want to do a frontal lobe like assessment, like a frontal polar EEG, then just stick one electrode like 3 centimeters above their right eye and one above their left eye, and then go for it. That wouldn't be great because people have vastly different head sizes. Even facial structure can differ massively between person to person, particularly if it's different, male versus female or different races. So what we do instead is we use the International 10-20 system, and these diagrams look like a hot mess, but they are just a way of showing you, like if we want to do a full EEG cap, like on the right here, there's a whole bunch of different points, and we can look at and compare any two of these different points, and that can give us a whole bunch of different useful information. The important thing to remember though is, so at the top here, we've got FP1 and FP2, which is an important one for medical and applied physiology. If you are doing that subject, you'll definitely come across those at some point. So these two are looking at frontal lobe EEG. And for example, they are comparing the signal between those two electrode points, and they are just measuring really large populations of neurons. So EEG is never able to look at structures deep within the brain. So it's only looking at like cortical activity, and it never is able to look at like small populations of neurons. It's looking at sort of really big areas, and that's sort of why the EEG waveforms look like they do. They're picking up on signals from lots and lots and lots, like literally millions of neurons at once. Is there much individual variation in there that can't be accounted for the percentage system? Yeah, so no system is going to be perfect. It is just a like pretty good standardized system, in the same way that you might say like BMI is not a perfect system. There are certainly large populations of people who don't fit within healthy standards of BMI, but they are sort of like objectively healthy. Proportion differences. Yeah, certainly. Yeah, certainly. There are lots of ways where the International 10-20 system will consider you like an outlier. So it might not necessarily, if you put an EEG cap on, it might not be like a perfect representation, but it's pretty good. It's really good. And it's really good that this is an internationally recognized system, because that way, if you do a study and you're just describing in your methodology, like, oh, like we used F7, F3, and CZ and C4, people know exactly what you're talking about. And they can repeat that study themselves, which is like one of the main scientific principles, is repeatability of your study and repeatability of results. So yeah, we don't worry too much about the fact that small individual differences can have a small impact on this. We just kind of do the best we can. All right, so your next question. The human cerebral cortex is very large and folded. What effect do these folds have on the brain signals that are recorded by an EEG? Yes. So we talked about in week one how, our first week of my tutorials, we talked about how you have your gyri and your sulci, and the brain is like spaghetti on a plate. So there's each of these gyri is like the noodle of the spaghetti. And one of the ways that it exists like that is to sort of increase the surface area of the brain. And that's, yeah, this question just wants you to understand why is that significant for EEG. So EEG measures the voltages generated by the currents that flow through synaptic excitation of the dendrites of many pyramidal neurons in the cerebral cortex. Wow, what a sentence. It just means EEG measures electrical activity of neurons. That's pretty much it. And it's really just looking at ones in the cerebral cortex, and even still, it's mostly just the surface level ones. So it's not able to look at the populations of neurons that are really deep. It simply can't happen. That's what this second paragraph is really trying to explain. So you've got your layers of the meninges, which, as you know, you've got 3 of them. You've got cerebrospinal fluid, or just fluid in general, bones of the skull, and skin. So this is all lies in between your electrode on the surface of the skin and between the neural tissue whose activity you're trying to measure. So lots of useless stuff that doesn't give off electrical activity that you don't care about, but it is impeding the signal. So that's basically why you can't measure the activity of like your hippocampus, for example, or just like the limbic system. Anything that's in those central areas of the brain, you absolutely would not be able to measure. And we have to use other sort of measures of understanding why they might be going wrong, or why there might be a pathology associated with those. Okay, we're going to look at an EEG demonstration. So someone attaching, it's me, me attaching an EEG to the wonderful David Van Rijk, who I helped film this with. So I apologize if you've already seen this before with David's subject, but I have to show it to you because I know not all of you are doing medical and applied physiology. Great.
Hi, welcome back to our math lab. So we're looking at electroencephalography, or EEG, and it is a way of measuring the electrical activity of neurons, so in particular large populations of neurons in the skin. And the way we measure this is by attaching electrodes to the surface of the skin. One thing I will point out with electrophysiology in general is when we're attaching electrodes to the skin, we're trying to pass a signal into this layer of tissue. So we have skin, tissue, sometimes we have bones in the way, and we also have hair in the way. So the way we deal with these sort of competing factors is we can add electrode gel, which increases conductance, and we can use metal caps on our actual electrode.
Video is very quiet.
This is a way to magnify or amplify that signal and its conductance, and it can give us a general idea of neuronal activity beneath the skin. Today, we're going to be looking at frontal lobe EEG activity. Let's jump into it. So here we have our electrode set up. We have our blue and our yellow electrodes with our metal caps, and these are our active electrodes. So these are going to be placed over our sites of interest, and they're going to be detecting large populations of neurons and their electrical activity. This black piece is our reference electrode, and we place it typically over the ear in EEG studies, because over the ear is a nice fleshy piece, and we're expecting little to no electrical activity. So the first thing we're going to do is we're going to find our two sites where we're going to place these electrodes to generate our frontal lobe EEG. And those are frontal polar 1 and frontal polar 2, FP1 and FP2. The way we find these two sites is using the International 1020 system. So this is a standardized...
Guys, if it's lagging, I'm just going to put the link up later for you guys to watch. Like this isn't even on YouTube. It's literally just, I've got the actual MP4 on my computer. So it's probably just my internet connection that's making it lagging. But if you are interested, I'm going to make sure the link is up later for you guys to watch. But yeah, I'm hoping for a large amount of you, it's still working just fine. So I'm going to keep finishing the video. But I am seeing that you guys, yeah, for some of you it is lagging. So I do apologize for that.
Internationally, and it is a way of separating the brain into segments that are compartmentalized based on 10 and 20% portions of the head. So FP1 and FP2, we're going to find them by measuring first David's circumference of his head, as well as the distance between his nasion, which is at the bridge of the nose, and his inium, which is the back, his bony piece at the back of the head. So we're going to start by taking some measurements. So to get our first landmark, we're going to measure from David's nasion, which is just above the bridge of his nose. We're going to run our measuring tape along the top of his head to the bony landmark at the back of his head, which is 30 centimeters. We're going to take 10% of that measurement, which is 3 centimeters, and we're going to go 3 centimeters above the nasion and make our first point. This is called FPZ, and FP1 and FP2 are going to be slightly to the left and slightly to the right of FPZ. So to get our next point, we measure the circumference of David's head, which is 55 centimeters. And we're going to take 5% of the circumference, which is 2.75 centimeters. Then we're going to go 2.75 centimeters to the right and 2.75 centimeters to the left. Now we have FP1 and FP2. And these are where we're going to attach our electrodes, and they are going to give us our frontal lobe activity EEG. All right, so we're ready to attach our electrodes now. So we have our two active electrodes, and we have our reference electrode. I'm going to get David to pinch this over his ear. You wouldn't mind, David? If at any point that becomes uncomfortable for you, just let me know. This white box here is our encoder. It's important for transmitting information that's picked up by the electrodes to the laptop. So I'm going to get David to put that in his top pocket just for convenience sake. This is our electrode paste. This is really important in ensuring that the metal plates of the electrodes attach properly to the skin and that there's good conductance there for the electrical signal to pass into the electrodes. So all I'm going to do is I'm going to scoop into each of these metal electrodes a bit of the electrode paste, scrape off any excess on the side of the rim, and then the electrode is ready to attach to FP1 and FP2. So we're going to use a bit of tape here to make sure that the electrode sticks properly to David's skin. And it can be quite helpful to loop the wire of the electrode around the ear, just to stop it from dangling in front of their eyes. So there we have FP1 and FP2. All right, so David has his up.
All right, we'll put it right there. So then I just look at like the actual numbers that get generated from the EEG. It's not really important for you guys to know. It's more important for MAP if you are doing that subject. But yeah, that's it. That's the demonstration.
Hi, welcome back.
No. Shut up. Aaron says my acting skills are a bit suspect. I don't know. Took me a couple of takes to get through all of that. All right, multiple choice questions. Yeah, I think actually to finish off today, we just have, oh no, we're going to do epilepsy. Okay, so there's a couple of multiple choice questions. And then we're going to get into epilepsy. Cool. So the EEG rhythm that is associated with a quiet waking state is alpha. Yeah. So just in like general, like relaxed state is you would predominantly expect to see alpha rhythms. 4 seizures are usually accompanied by very large EEG patterns. We haven't talked about this yet, but it is true. In fact, in general, when you see like very large means amplitude, right? So in general, if you see really large EEG patterns and that person is not asleep, it's generally pathological. It's not healthy to have large EEG patterns because that is representative of synchronous brain activity, and healthy brain activity is asynchronous. It's all over the place. Your neurons are really playing it fast and loose. In general, what part of the brain's electrical activity is measured by an EEG? That one's easy. It's just the cerebral cortex. We're not able to really measure these other 3 here. Okay, let's move on to epilepsy. So epilepsy is the main pathology that we're going to be looking at today, which is really cool. I think it's the first kind of actual like pathology that we spend a fair bit of time on, aside from like the hydrocephalus from week 1. So epilepsy is, be really helpful if you guys could remember just like a general definition of it, but it is a transitory disturbance of brain functions. Transitory meaning it can change, it can shift from being sort of localized to widespread, and it's generally temporary. It's not a permanent, like you will not be in a permanent epileptic state. It's a state of change. It is a serious neurological condition with a peak incidence in early childhood and in the elderly. So if you look at incidence by age, it looks like a U. So you by age. So children, highly affected, and then in the middle of your life, not really seeing that much of an incidence, and then you see another peak incidence as you grow into an elderly. Various types of epilepsy, which we'll get into. About 2/3 of epileptics have no obvious cause for their seizures, and about 2/3 of them will stop having seizures within 2 to 5 years of their onset, usually in the context of taking medications. So medications are, I guess, they're in a reasonably good place right now. 2/3 stop having seizures within 2 to 5 years. Yeah, I think that's, obviously we want to improve on every asset or every aspect of medical science, but that seems to be in a reasonable place right now. Seizures can be incredibly debilitating, and there's a range of situations where people with this neurological disorder, they just have to make such drastic life changes. For example, even driving for some people with severe epilepsy, even just trying to drive to the shops can become a hazard, because if you have a seizure while you're trying to drive, you know, you're going to cause an accident. Swimming, there are people who have been like almost Olympic level swimmers who have then developed epilepsy. Again, it's incredibly rare outside, well, it is rare outside of childhood and the elderly, but it can happen. And people who, you know, might like swimming or just going to the beach in general, now becomes a drowning risk if you have an epileptic seizure. And depending, yeah, it really depends on the type of epilepsy that you have. Many different types. Medications can have various different effectiveness between person to person. And yeah, so treatment is based on modifying the balance between inhibitory gamma aminobutyric acid, or just GABA, and glutamatergic network within the brain, or the repetitive firing potential of neurons. Sorry, I should say brain. The EEG recordings reveal that epileptic events, which we refer to as ictal events in a couple slides as well, they're associated with either generalized synchronous or focal spike and wave discharges. So that is your description of an EEG waveform for epilepsy. These events can also be seen transiently at other times without a seizure or interictal activity. So inter means in between. So interictal just means between actual seizure events. So what does an EEG actually look like? We have a diagram here on the right. So So you've got 6 electrodes that are hooked up here. I think LO and RO are left occipital and right occipital. LF and RF are left frontal, right frontal, and then LT and RT are left temporal and right temporal. So you've just got 6 electrodes placed at really general parts of the brain. And as you can see, in all areas in this particular ictal event, the seizure, you see these really high amplitudes. So these really indicating really like widespread synchronous activity of neurons. This huge paragraph here is like, it's just giving you like a really wordy explanation or description of what an EEG looks like, and sort of the molecular background that's happening with them. So interictal activity corresponds to a depolarizing shift with superimposed action potentials from an assembly of neurons. Sounds good so far. Hyperpolarization follows as local inhibitory interneurons are activated. Repeated interictal spikes lead to the period of hyperpolarization shortening. So that's where you have, after depolarization, you have your hyperpolarization phase, where the individual neuron will become more negative than its resting state. So it has hyperpolarized itself. And if you shorten that period, then you shorten your absolute refractory period. So remember from week 1, your absolute refractory period is a period where, it's shortening your relative refractory period, which is the period after your main depolarization, where your neuron can be activated again, but only if you have a greater stimulus. So the shortening of that does sort of, has like a positive feedback almost, and it continues with this recruitment of action potentials in this localized population. But it can turn from a localized incidence into a widespread thing. So to answer your question here, don't some epileptic seizures only affect parts of the brain? Yes, that's absolutely true. So we're going to look at the difference between partial seizures, which only affect maybe like one lobe or one cortex of the brain, versus generalized seizures, which affect the entire brain. Where did I get up to? And this activates ion channels in the neurons and increases extracellular potassium concentrations, which further depolarizes the neurons. So that whole 2 sentences there is just explaining how populations of neurons continue recruiting new populations of neurons, and you just get this widespread, like if one person starts screaming in a park, and then that causes more people to start screaming, and then soon everyone in the park is all screaming at the same pitch at the same time, and it's just absolute noise. That's kind of, I guess, a terrible analogy for what a seizure is. If sufficient neurons are activated, then synchronous discharges are produced in many neurons, which can lead to a seizure. Yep, synchronous discharges, I guess, is one of the common things you're seeing here that is associated with epilepsy. So seizure is then terminated by ion channel inhibition within the neuron and within neural network by GABA-related interneuronal activity. Cool. Is it important to remember all this information? It used to be. I would say, you know what, I would have this on hand during your exam. I mean, you're going to have, it's open book, so you can have all your sort of lecture slides open. If you get a question that is related to any of this, you know exactly where it is. But in terms of understanding it, I would say the main takeaway from this is understanding that, or how understanding how a seizure results from this hyperpolarization shortening, increase in extracellular potassium concentrations. There's a few like keywords here that I would say are important, but it's one of those things where I've heard it described as like all the traffic lights being on at once. Yeah, for sure. Yeah, there's another great analogy. It's just important for you to recognize that all neurons activating at the same time is very bad. It is a pathological state, and a seizure is more or less a symptom of that. And epilepsy is like chronic seizures. It's like having repeated seizures. And there is just a wide range of sort of causes for it. We really don't understand the pathology of epilepsy well enough, or even seizures in general. All right, we are still on this. Oh, okay, so I'm just describing what's happening at each point. So at number 1, this is before your seizure. So for each of these, I know left temporal and right temporal look like they're flatlining a little, but that is normal for temporal activity during a waking state. So all 6 of these are just representing a normal like beta rhythm. So it's before a seizure. At number 2 is the onset of your seizure. So widespread synchronous activity. 3 is your clonic phase, which I'll explain in a little bit, in which the person makes rhythmic movements in time with a large abnormal electrical discharges, which is very interesting. So you can see 1, 2, 3, 4 of these sort of rhythmic spikes, and they are directly correlated with rhythmic movements in the person's limbs, for example. And then 4 is this sort of like comatose state after the seizure ends. So anything else I want to say about that? Nah, pretty good. All right, so just a couple of questions on epilepsy. Yep, so we already defined epilepsy as a transitory disturbance of brain function that develops suddenly and can cease spontaneously. Most prevalent during childhood and the elderly. What is the common non-invasive way to measure epileptic fits? It is EEG. Yeah, to my understanding, all the types of electrophysiology that we study at UTS are non-invasive, so it's just using like gel attached to the skin. And they're associated with either generalized synchronous or focal spike and wave discharges. This abnormality can also be transient. So what is the cellular physiological model of epilepsy? This is almost cut and paste of what we already looked at before, so I won't go through it again. But yeah, because what we used to do, we used to have open book, for example. We used to have open book exams and open, like almost like essay style. But we wouldn't ask you to write like essay style, like long extended response is the word I'm looking for. So you could be expected to be able to not regurgitate necessarily, but be able to have dot points for all of these in your answer. If we asked you what was the cellular physiological model of epilepsy. So I would just, if you see any question that is related to this and you really don't feel like going through this entire slide and revising it, because I know it looks quite intimidating, then you know exactly where to come and look for this. So more questions. Seizures are usually accompanied by what? Very large EEG patterns of about 3 Hertz. So 3 Hertz is less than 4. As we know, less than 4 Hertz is typically your delta waves, which you would see during slow wave sleep. I don't know that we necessarily categorize seizures as delta waves. Technically, they might be because they are less than 4 Hertz. I'd be very surprised if they actually were. They almost seem to be their own like category because they are very large EEG waves. 5, a generalized seizure involves the entire what? It is the cerebral cortex. Why do EEGs with relatively fast frequencies tend to have smaller amplitudes? So that is a trend that we saw where going from beta to alpha to theta to delta is that you would start with really fast frequencies and small amplitudes, and then you would go to the opposite. So why is there a tendency for that to happen? And we've kind of talked about it already, but it's really just to do with that overlapping activity of different neurons. So if you have a group of neurons that are excited simultaneously, their individual signals are going to start overlapping. And an EEG is measuring the sum of all those different neurons firing. So one signal of millions of neurons is going to look different. It's not going to look anything like an individual neuron's activity. That's kind of the whole lesson I've tried to explain today. yeah. EEGs with relatively fast frequencies tend to have smaller amplitudes. That's the main takeaway from this. That's all I'd be worried about. List some common causes of seizures by age. So this, we talked about how there's a high incidence in infancy and in the elderly. The best way to remember this diagram, if you do get a question on it, is that the major causes for each age range are just the things that happen, or I guess are most at risk for that age group anyway. So like infants, for example, their major causes are birth injury, hypoxia during birth, ischemia, congenital malformations, and infection. Yeah, infants are at risk of infection. So these are things that children are already at risk for. So it kind of makes sense that these are also causes for seizure. Because by the way, these are all really broad causes. These are really just, it's really just stress. It's really just physiological stress on the body seems to be the main thing that is responsible for causing seizures and also epilepsy. So if you want to sort of remember what each of the major causes are by age group, it's really just what is the main source of danger for that age group. And if you look at young adults, it introduces drug use, which is obvious because young adults are experimenting with drugs, alcohol, yep. Head trauma might be common in young adults because they get into car accidents more often, or they're starting to play contact sports and that kind of thing. Head trauma is also common to childhood. And then elderly, yes, so strokes, brain tumor, cardiac arrest, metabolic origin, yeah, maybe if they have diabetes or something like that. So again, these are all things that you would expect to see of a high incidence in the elderly. And so they are also coincidentally related to causing seizures in that age group. Okay, so now we can talk about the different types of seizures. So simple partial seizures, and we have complex partial seizures. So simple is where we have one hemisphere or lobe affected. The person or the patient will remain conscious throughout the seizure, and it's just involving a limited region of the cortex. So it can manifest as a motor impairment or a sensory impairment. So maybe they will see, they might lose their vision, or they might see flashing lights. They may have emotional or hallucinatory phenomenon, such as deja vu, which is sort of a really overpowering deja vu they might have. And it can last from a few seconds to a few hours. So that is your simple partial. And your complex partial seizures is where you have one hemisphere or lobe affected, but there is a loss of consciousness, or at least impaired consciousness. So, but the common thing between partial seizures is that it's just affecting one specific area. And then we'll look at generalized seizures in a minute. So altered consciousness is due to bilateral spread of the seizure discharge, and the patient will have no recollection. May begin with or without a warning or aura, or with stereotyped motor, sensory, autonomic, or psychic symptoms. Lasts for a couple of minutes, and it can be followed by a period of fairly long confusion. All right, generalized seizures. So this is where you have both hemispheres are affected and definitely have loss of consciousness. So when you have such widespread synchronous discharges, you're going to lose consciousness. It's just simply not possible for the brain to continue acting or functioning normally when you have just the entire brain is undergoing a seizure. So you lose consciousness, and you will see a range of symptoms. So tonic seizures are where you have your muscles stiffen and flex. Clonic is characterized by these convulsions. And then you can have tonic phase intermediated by a clonic phase. So they kind of interchange. Atonic seizures are where your muscles relax. Myoclonic is where you have short muscle twitches. And then you have absent seizures, which are kind of cool. Absent seizures are also called petit mal seizures, and they are pretty common in children. So patients are between 3 and 20 years old. I've seen videos of these online. They're very fascinating if you want to go look up a petit mal seizure, but it is where you have like a child will just be sitting, maybe talking about their day or something, and they will just rapidly just switch off. Their face will kind of go blank. You might have these automatisms like eye blinking or lip movements, followed by increase or decrease in muscle tone, mild jerks. Largely though, it will just seem like they just stopped in the middle of their conversation and they just start staring into space. And it usually only lasts around 10 seconds, and you can try clapping in their face, you can try getting them out of it, which I wouldn't really recommend because it's only a short-term thing, but they will be completely unresponsive. And to a lot of people, it just seems like they're losing concentration. So it just seems like they've zoned out. To like parents and teachers who might not be aware of this being a pathological condition. And the really crazy thing is that a child might be just talking about their day and then they switch off. 10 seconds later, they switch back on and they just continue mid-sentence without even realizing that they lost consciousness, which is pretty crazy when you think about it. And it can go misdiagnosed or not even picked up on for a really long time. But generally, it's not too much of a danger. Recovery tends to be immediate, but they'll have no recall of losing consciousness or anything like that. Is it possible for someone who is having a simple partial seizure to not realize they're having it? So let's go back to simple partial. I don't know of any reason why they wouldn't know. I would say that they're probably aware of, they would have symptoms that they are aware of, that they are sensitive to. So generally they should know. I guess though, what you're asking is it possible for someone? Potentially, because they still remain conscious. They can manifest as motor impairment or sensory impairment. Yeah, there may be very rare cases where it is technically a simple partial seizure, but they just don't realize it. Maybe they're just too busy to realize that they were hearing things or something like that. But for the most part, yeah, it should manifest as something that you are sensitive to and aware of. But yeah, very, very interesting. Okay, this slide. Wow, I hate this slide. List some anticonvulsants used to manage seizures, their mechanism of action and side effects. So again, I would put this slide on list of slides to not really care that much about. Just be aware that it exists, and technically we can ask you a question about mechanisms of action for certain anticonvulsants. You can see that 3 of these kind of have, 4 of them all act in the same way. They inhibit voltage-dependent sodium channels in a voltage and use-dependent manner. That's one of the main themes of the mechanisms of action for anticonvulsants. So these are just used to manage or treat seizures. So I would know that much, that they all share this main mechanism of action, except this last one, which blocks voltage-dependent calcium channels. I would also be aware of some of these side effects, and I've got the definitions for some of these on the left here. So for example, ataxia. Don't really expect you to know that, but it is a disruption of muscle coordination. A couple of other side effects that I've got there. Yeah, and then in terms of just do you need to remember like the phenytoin is used to treat generalized seizure? I wouldn't go out of my way to remember it. I would just be aware that this slide exists, and because you have an open book exam, you can come back to this at any point, and you know exactly if any point you come across a question that mentions anticonvulsions or medications used to treat epilepsy, this is exactly where you need to come. Cool, awesome. We are done for that. I had a question last week about, let me try this again. had a question last week about questions that I had for revision for week 1 that I'm going to go through real quick. So these are the questions, if you remember them. Let me press some buttons. I'm just going to leave it on this screen, actually. Okay, so the first one was, what are the 3 layers of the meninges? Here they are. We already went through them, so I'm not going to spend any extra time on this. But if you're watching this not live, you can just pause the screen here. Where is the cerebrospinal fluid produced? So it is produced in a special tissue called the choroid plexus. By the way, I just got all these answers straight from the textbook. So I don't expect any of you to have the textbook. I guess that's why I'm showing you these answers, because technically they are revision. I don't really see them come up that often, but they complement the content that I delivered anyway. So I'm going to go through them anyway. Called the choroid plexus in the ventricles and the cerebrum. They exit the ventricular system and enter the subarachnoid space. So remember, we talked about that space between the layers of the meninges. They do so by way of small openings located near where the cerebellum attaches to the brainstem, and they're absorbed by blood vessels at certain structures called the arachnoid villi in the subarachnoid space. So that is just a little bit more information on cerebrospinal fluid. 3 was, where do the cranial nerves arise from and what do they innervate? So they arise from the brainstem and they innervate mostly the head. That's all I could find on that in the textbook. And yeah, so this last question was weird because I couldn't find anything in that whole chapter on parasympathetic axons. And it doesn't really make sense. I think what happened was the original person who made these questions saw the abbreviation PNS and thought parasympathetic, but that's not true. PNS is 100% peripheral nervous system. So I've just reworded this question to say, where do axons of the peripheral nervous system originate from? And that just makes way more sense because that's what your actual chapter in the textbook is on. So your peripheral nervous system, as I mean, earlier in this, I think I did have, or maybe it was the week after, but I talked about like the different branches of your nervous system, and your peripheral nervous system has a somatic component, which is related to mostly muscles, but also some sensory stuff as well. And you have your autonomic nervous system, which is basically really broad, but it's mostly involuntary processes. And so this question, how I've interpreted it, it's only concerned with your somatic nervous system. So somatic sensory axons come from your dorsal root ganglion. This is the diagram that we had earlier in that slide, presentation. So you have your dorsal roots and your dorsal root ganglia and your spinal nerve. So there are dorsal root ganglia for each of your spinal nerves. At your dorsal root ganglia, which are outside the spinal cord and thus part of your peripheral nervous system, this is where your axons enter the spinal cord, and anything that's coming into the spinal cord is known as an afferent axon. I apologize if this is getting like way off topic for you guys, but this is really important stuff. So I just want to make sure that you have all the information here. Okay, so for example, carrying sensory information from the skin to the central nervous system, and it doesn't have to go to the brain. So for example, if you put your hand on a hot plate, like a grill, that reflex arc is going to your spinal cord. It does not need to be processed by your brain. The sensory information goes to your central nervous system in your spine, and then it bounces straight back to your hand, and you pull your hand away from the hot plate. Doesn't need to go to the brain. You don't need to think about, should I take my hand away from the hot plate or not? It's just an immediate reflex. So that's your somatic sensory axons. So they're always going from the peripherals to the spinal cord. So they enter the spinal cord, and your somatic motor neurons actually derive from your ventral spinal cord. So technically, some parts of these axons are in your spinal cord, so they're in your central nervous system, but for the most part, their axons are sitting outside the spinal cord, so they are mostly in your peripheral nervous system. And these axons are exiting your spinal cord. So they're taking information from your central nervous system and they're taking it outside of your spinal cord to your peripheral nervous system to your muscles, like to do work. So for example, commanding a skeletal muscle to contract. So following on from that example, your dorsal root ganglia will take information of my hand is on a hot plate, and then your ventral spinal cord will then rebound that signal and say, oh, contract your biceps, move your arm away from the hot plate. And it's a very quick movement of information. And there is a dorsal root ganglion for each spinal nerve, and they lie outside the spinal cord. So just a little bit of additional revision there for week 1. I apologize that that is a bit of a backtrack. Yeah, you guys will backtrack to this a lot anyway as well, because you'll be doing your anatomy pracs throughout the semester, which I hope have been going well for you guys. So that's everything for this week. That's all I wanted to say. And it's 4:01. Awesome. Thanks, guys. See you again next week. Enjoy your weekends, and we'll be picking up again next week. Thanks for coming, everyone. Enjoy. See yous.